Okay, hello everyone. My name is Naseel, and today I will be taking you through fatty acid synthesis. All right, so this is my contact information if you need to ask me any questions later on. All right, so let's start. So first of all, we'll be talking about fatty acids in general. We'll be giving you guys an introduction to fatty acids. And then second, we'll be talking about the fatty acid synthesis itself, which occurs in 3 steps. Okay, now this will be the main bulk of the lecture. All right. Third, we'll be talking about triglycerides very briefly. And fourth, we'll do some practice questions together. All right. So let's start. So the question we always ask ourselves is, do we really need to eat fatty foods? Like, you know how they're essential proteins that we need to obtain from diets? Well, is that the same with fats? Okay. I mean, look at the burger and the desserts. These look really good. Okay. So the answer to this question is it's a yes and a no. Why do we say that? Because we have non-essential fatty acids that we can make in our body, so we do not need to obtain them from diet. However, we also have essential fatty acids, meaning that we cannot make them in the body and we do need to eat them, okay? However, that doesn't mean you're allowed to eat those burgers, okay? These essential fatty acids come from so-called healthy oils, okay? So things like salmon, olive oil, walnuts, all right? But those desserts and burgers, they're not that good, okay? So let's talk about fatty acids and how to name them. Here we have our main fatty acid, palmitic acid or palmitate, okay? It's a 16 carbon fatty acid. So if you count the carbons, you'll find that they are 16, okay? Another thing you need to know about fatty acids is that we have two ends. First, we have the COOH group, which is the carboxylic end, okay? And on the other hand, we have CH3, or the methyl end, okay? And this is where the concept of omega comes from, and we'll talk about that, okay? Now, how do we name fatty acids? Fatty acid is named by a ratio, okay? So it's the ratio of carbon atoms to the number of double bonds that we have counted from the carboxylic end, from the COOH group. However, do we have any double bonds here? No, okay? So the name of palmitate is going to be 16 to 0, okay? And this means that palmitate is saturated. Remember that saturated means we do not have double bonds, they're only single bonds, okay? Another thing to note about palmitate itself is that we can only synthesize it, fatty acids up to carbon number 16 using the fatty acid synthase enzyme, okay? I'll be shortening it for FAS, okay? And FA, by the way, stands for fatty acids, okay? Now, let's take this a step further, okay? We have stearic acid, all right, which is 18 to 0. What does this mean? This means we added 2 more carbons, okay? However, do we have a double bond? No, because it's still 0, okay? Can we make stearic acid in the body? Yes, okay? So this is not an essential fatty acid. Moving on to oleic acid, okay? Here we notice it's 18, okay? But we have 1. What does that mean? It means we have 1 double bond, okay? You can see it right here, all right? Now, something else is that we put between parentheses the position where the double bond is. So it's 9, carbon number 9. Where do we count it from? Like we said, from the carboxylic end. So you go 1, 2, 3, 4, 5, 6, 7, 8, 9. Here, it's carbon number 9. We have a double bond, okay? And this is oleic acid. Oleic acid, since it has a double bond, it is an unsaturated fat, okay? Meaning that it has a double bond, all right? Can we make oleic acid in the body? Yes, again, okay? Stearic acid and oleic acid are made in the body too, but they're not using the fatty acid synthase enzyme. They're using other enzymes, and we'll name them later on, okay? Moving on, we have linoleic acid, okay? Here we can see we have 2 double bonds, one at position 9, one at position 12, okay? Now, here, starting off from linoleic acid, and down, we cannot make them in the body. Okay, so linoleic acid is an essential fatty acid. Okay, same thing, linolenic acid, which has 3 double bonds, one at position 9, 12, and 15, okay, cannot be made in the body. So these two are essential fatty acids. Okay. Moving on, we always talk about omega-3, right? We hear it, we can buy it, okay, as a supplement. Now, what is omega-3? Omega-3 means that you're counting the double bond, but from the opposite direction. So, right, we said we usually count from the carboxylic end, from the COOH. Now, flip the switch, okay? You go from CH3, the methyl end, and you count, okay? So, for example, 1, 2, 3. So this is omega-3. Linolenic acid is omega-3. Okay? Same way, linoleic acid would be 1, 2, 3, 4, 5, 6, omega-6. Okay? Here I've written this down. Omega bonds are counted from the methyl end from the opposite direction. So linolenic acid, which is the bottommost one, this one, is omega-3, and the one above it, linoleic acid, is omega-6. Okay? Now we can synthesize stearic acid and oleic acid through different enzymes. Stearic acid is synthesized through an elongase, from the name elongase meaning that it elongates the fatty acid chain. Okay? Oleic acid, since it has a double bond, we need to desaturate it. So it is synthesized by a desaturase. Okay? Now, this process occurs in the endoplasmic reticulum in the ER, unlike the normal fatty acid synthesis, which occurs in the cytoplasm. Okay? So, is this concept clear? Yes. All right. Okay, then we'll go to the next concept. Okay, so let's just talk a bit about fatty acids. They're synthesized in the liver, adipose tissue, and mammary glands. Okay, and fatty acid synthesis occurs in the fed state. Now, what do I mean by fed states? It means that you have eaten way too much, okay? Your body doesn't need to produce more energy, okay? And it says, that's it, that's enough. I'm going to store all this energy in the form of fat, okay? Because we know that when we eat, glucose goes through glycolysis, then TCA cycle, and then electron transport chain, oxidative phosphorylation, all that to produce energy. Okay. However, if we've eaten way too much, we do not need to produce more energy anymore, right? So we resort to storing this energy as fat. Okay. Now, we lack the enzyme needed to form double bonds at omega-3 and 6, like we've explained. Okay, so basically we cannot form bonds past position 9. Now, when I say position 9, I'm counting from the normal end or the COOH end. Going back here, so as you can see, we were able to form oleic acid, and it is at carbon number 9. But we cannot go to carbon 12 and 15 in the body. We need to obtain these from food. Okay? So these essential fatty acids, linoleic and linolenic acid, must be consumed. Okay? And these themselves, these two fatty acids, are precursors to other fatty acids that have omega-3 and omega-6 bonds. Okay? Final thing is that deficiency of these fatty acids leads to retarded growth and skin lesions, but this is not really. Now, omega-6, okay, is a precursor for another fatty acid, which is arachidonic acid. Okay, now this one's very important and we'll say why later. So arachidonic acid itself is a 20 carbon molecule. Okay, it is omega-6 meaning that it's derived from linoleic acid because we said linoleic is omega-6 and linolenic is omega-3. Okay, this arachidonic acid is a precursor for signal molecules called eicosanoids. Now, eicosanoids, it's a weird name, okay, but we will explain them in the next slide. Okay, they're paracrine molecules, meaning that they're short-lived and they only affect nearby cells, meaning where they are synthesized or where they are produced, that is where their action would be. They wouldn't go away and act on different parts of the body. Okay? Now, here, this is a figure showing arachidonic acid and its products. But let's start here. So we have arachidonates or arachidonic acid, they're the same thing. This can come from two products, okay? Two things. Diacylglycerols or phospholipids, okay? So diacylglycerols are broken down by diacylglycerol lipase, and then you get arachidonates. Where phospholipids are broken down by phospholipase A2, you get arachidonates, okay? So we're done with that. Now, what does arachidonate itself produce? First, it produces prostaglandins, which is very important, okay? And you'll hear it a lot, all right? So we get prostaglandins. Then these prostaglandins have different types, okay? First, we have prostacyclin. Okay, let's talk a bit about prostacyclin. It's a vasodilator, meaning that it makes blood vessels wider, okay, to improve blood flow, okay? Also, it inhibits platelet aggregation. What does that mean? We know that platelets are involved in clotting, right? So if you inhibit that, that means you inhibit clotting. Now looking at thromboxane, okay, this is the opposite of prostacyclin. It does the opposite. So it's a vasoconstrictor, meaning it narrows blood vessels, and it promotes clotting, okay? So you can see that our body has a balance, okay, between these two things. Because let's say I only had prostacyclin, theoretically, okay? I would have inhibition of platelet aggregation, I would have no clotting whatsoever. So if I get a wound, a scratch, it won't heal, it will keep on bleeding, okay? On the other hand, if I only had thromboxane, that means a, I have a predisposition to clot, okay? So that means I'm at risk of what? Strokes or heart attacks, okay? You guys have done CVP already, you know that if you get a heart attack, you know what happens next, right? There's no blood flow, you get ischemia, correct? So our body has a balance between these two things. Other prostaglandins cause inflammation, pain, fever, okay, all these things. And something significant is that it can induce birth. That's why sometimes in hospitals, we give women who are in labor, we give them prostaglandins, okay, so that you can induce labor, okay? So this is the first product of arachidonic acid. Now another product of arachidonic acid is leukotrienes. Leukotrienes are the culprits behind asthma, okay? Why do I say that? Because leukotrienes are bronchoconstrictors, meaning that the bronchioles and the bronchi become constricted. So breathing becomes harder. So for people with asthma, leukotrienes basically cause this bronchoconstriction. And so that's why you can give medications that act against leukotrienes. For example, if you guys know anyone who's asthmatic or you yourselves are asthmatic, you might be taking Singular, okay? That's what's given here in Saudi Arabia, all right? So this is all about arachidonic acid. Is it clear? Do you guys have any questions? Okay, okay, moving on. So that was all about fatty acids in general. Now we will go into the main thing, which is the synthesis itself. Okay, so this is an overview. We'll briefly talk about the three steps. First, we need to deliver the acetyl-CoA from the mitochondria to the cytoplasm. Okay, so I know acetyl-CoA is produced in the TCA cycle, right? Now we need to take that from the mitochondria to the cytoplasm where fatty acid synthesis occurs. To do that, we need citrate. Okay, we'll explain this later. Okay, step two is that after we've taken acetyl-CoA outside, we need to convert it to malonyl-CoA. Okay, and step three is the actual synthesis of the fatty acid by the fatty acid synthase enzyme, where we keep on cyclically adding fatty acids on top of each other, carbon molecules on top of each other, until you get the final 16 carbon fatty acid chain, which is palmitic. Okay, so before we go into step one, let's talk about the ingredients you need to make fatty acids. So we need acetyl-CoA, obviously, because it carries the carbon, right? Then you need to convert it to malonyl-CoA. But to do that, you need carbon dioxide and you need ATP, okay? Then last thing, you need NADPH. Now, what does NADPH have to do here, okay? NADPH has hydrogen atoms, right? So it can be converted into H plus, right? H plus has a reducing power, meaning that it adds hydrogen ions on anything, okay? We need this later on when we will see why, okay? So all these ingredients are put into this red bowl, and then the enzyme here that mixes everything is fatty acid synthase, okay? Now, let's go through step one. First, glucose is converted to pyruvate through glycolysis. We already know this, okay? Up till now, this is what normally happens in the body, right? Then pyruvate goes into the Krebs cycle. Basically, it's transported from the cytoplasm into the mitochondria, and then it's converted either into acetyl-CoA by pyruvate dehydrogenase, PDH, or into oxaloacetate by PC or pyruvate carboxylase. These are the names of the enzymes. Okay, now let's go through the Krebs cycle, which I'm sure you guys know more than I do. Okay, so acetyl-CoA and oxaloacetate combine to form citrates, and acetyl-CoA loses the CoA group. Okay, remember this. All right, citrate goes to isocitrate, isocitrate goes to alpha-ketoglutarate, and then to succinyl-CoA, and so on. Okay, normally this cycle goes on. All right, and in this cycle you produce ATP, NADH, and FADH2. However, like we said, because you've taken in way too much food, okay, this cycle has run way too many times, and so you produce so much ATP and so much NADH. If you guys remember, these two things inhibit enzymes in the TCA cycle. The enzymes that are inhibited are alpha-ketoglutarate and isocitrate dehydrogenase. So, if I inhibit these two enzymes, what happens to isocitrate, which is right here? It accumulates, okay? But also, citrates accumulates, and it accumulates much more than isocitrates. Okay, you'll tell me, how does citrate accumulate if isocitrate is the step right before the enzyme inhibition? Well, this step from citrate to isocitrate is reversible, okay? So isocitrate gets pushed back to citrate, all right? And then citrate goes and leaves the mitochondria, okay? I've written this down again. So because we've already produced so much ATP and NADH, these two enzymes are inhibited, and so isocitrate and citrate accumulate, and citrate leaves the mitochondria. Is this part clear? You guys want me to repeat it? Okay. So we said citrate leaves the mitochondria, right? So we're back in the cytosol, okay? I've taken this picture from your slides, your lecture, but trust me, this was the best there is, okay? So we'll go through it in details. So citrate, now it's outside. It's in the cytoplasm where we need it to be, okay? It's broken down into acetyl-CoA and oxaloacetate, okay? The enzyme that catalyzes this reaction is ATP citrate lyase. Please remember this one, okay? It's very important. Now, what do we need for this enzyme to work? We need a CoA group, obviously, because we said that acetyl-CoA lost its CoA group. Look here, acetyl-CoA, and then it loses the CoA. So we need to add it back because we're going to break it down into acetyl-CoA and oxaloacetate, right? So we need the CoA group, and you also need ATP because the enzyme literally says ATP citrate lyase, so it needs ATP, okay? Then acetyl-CoA goes into step 2. Okay, so we won't talk about it right now. However, oxaloacetate does something interesting. It goes and becomes converted back into pyruvate. Okay, how does it do that? It goes to malate first by malate dehydrogenase, and then malate gets converted to pyruvate by malic enzyme. Okay, you'll ask me, why did we need to go through all this just to come back to pyruvate? Okay, didn't we start off with pyruvate here? Why do I need to go through all that and Krebs cycle and take it out and then go back to pyruvate? Because first of all, you need the acetyl-CoA for the carbons, okay? And there is no way to take acetyl-CoA out of the mitochondria except through citrate. Second, we have this cofactor, Remember, we said we need NADPH, right? Now, the only way we can produce that is through this pathway, okay? Especially through malic enzyme, okay? So, stage 1, okay? So this is basically the main place where we say, okay, this is stage 1 of fatty acid synthesis. What happened before are the steps leading to fatty acid synthesis, okay? So, first, citrate is broken down into acetyl-CoA and oxaloacetate by ATP citrate carriers, and then oxaloacetate is converted to malate to pyruvate, and this produces NADPH, okay, the cofactor needed for fatty acid synthesis. Now, this might ring a bell. Where else did we see NADPH? I'm not sure if you guys took it, but does anyone know? Does anyone have a guess of where else we can get NADPH? EPP, good job. Okay, pentose phosphate. Okay, so let's go through it briefly, very briefly. Okay, first we have again citrates broken down into acetyl-CoA and oxaloacetate. Acetyl-CoA goes into fatty acid synthesis. Oxaloacetate goes to malate to pyruvate, and this produces NADPH. Now look at this side. We have glucose, and the main idea of the pentose phosphate pathway is to convert glucose to ribose 5-phosphate. Okay. Now, ribose 5-phosphate, if you guys remember, it is the sugar involved in RNA. Okay. So basically, this pathway, the pentose phosphate pathway, is involved in DNA synthesis. Okay. Either way, this pathway produces NADPH as well. Okay. And this is put into the fatty acids. Okay. So this is a summary of step 1. Goes to pyruvate through glycolysis. And then pyruvate goes to the mitochondria. Pyruvate goes to acetyl-CoA to citrate. And then citrate goes back to the cytosol. It's broken down into acetyl-CoA and oxaloacetate. Acetyl-CoA goes into step 2, which we'll talk about now. And oxaloacetate goes to malate, goes to pyruvate. And then this releases NADPH. Okay? So I've highlighted the 2 things that we got out of step 1. Okay, the 2 ingredients. So is step 1 clear for everyone? Okay, good. Alright, let's go to step 2. So step 2 basically involves this main enzyme, ACC or acetyl-CoA carboxylase. Okay, this is the main player in step 2, and it's very important. So remember we said we want to convert acetyl-CoA to malonyl-CoA? Now look, acetyl-CoA is a 2-carbon molecule, while malonyl-CoA is a 3-carbon molecule. So in order to do that, we need to add a carbon to acetyl-CoA. In other words, carboxylate it. So that's why it's called acetyl-CoA carboxylase. Okay? And we obviously also need the carbon dioxide molecule to get the 3rd carbon from. Okay? Remember, this enzyme requires biotin, which is a vitamin. If you guys really want to know, it's vitamin B7, okay, but that's not important. And it also requires ATP. This enzyme is very important. Why? Because it is the rate-limiting step of fatty acid synthesis, okay? If there's anything you guys need to remember about this lecture is that acetyl-CoA carboxylase is the rate-limiting step of fatty acid synthesis of the whole process, okay? So, see what we've written here. Acetyl-CoA is carboxylated to malonyl-CoA. Then this enzyme, acetyl-CoA carboxylase or ACC, catalyzes this reaction. It is the rate-limiting step in fatty acid synthesis. Okay? This is very important. Please remember. Now, acetyl-CoA carboxylase, like we said, requires biotin. However, it's dimeric, meaning that it has 2 portions, okay, stuck together, and it can be regulated. I mean, usually all rate-limiting steps are regulated, right? So this is just another one. Now let's talk about the biotin part for a bit. So, this whole thing is the enzyme, okay, acetyl-CoA carboxylase. It has 3 parts: biotin carboxylase, transcarboxylase, and this biotin carrier protein, which carries biotin. So this molecule is biotin, okay? First step is that biotin gets carbon dioxide attached to it, and then biotin carboxylase activates this carbon dioxide. Okay, so this is the first part of this enzyme working. Okay. Second, this biotin carrier protein swings the biotin to the other side of the enzyme. It swings it towards the transcarboxylase. Okay, specifically the active site of transcarboxylase. Okay, what does transcarboxylase do? It adds this activated carbon dioxide to acetyl-CoA. Okay, so so the biotin now has the carbon dioxide attached to it. Biotin gives this carbon dioxide to acetyl-CoA. So what would you get out of this? Melanin-CoA. You get 3 carbons. All right. So again, ACC has a biotin carboxylase site, which activates the carbon dioxide on the biotin carrier protein. Okay. Step 2, the biotin arm swings this activated carbon dioxide to the other side. Okay, from biotin carboxylase to transcarboxylase. Okay. Then 3, transcarboxylase gives acetyl-CoA this carbon dioxide. Okay, and you get malonyl-CoA. All right. So, is this part of biotin clear? It might probably be the most complicated part. Can I repeat? Would you like me to repeat? Okay, no problem. I'll repeat. So, we have, again, let's start over. We have this enzyme, ACC, okay? It has 2 parts, biotin carboxylase and transcarboxylase, okay? If you guys notice the blue color, that is where we're saying that, okay, this part of the enzyme is active now, okay? And another thing you need is the biotin carrier protein, okay, which has biotin attached to it, all right? Now, the biotin itself on the biotin carrier protein is leaning towards biotin carboxylase now. This biotin carboxylase adds a carbon dioxide molecule onto the biotin, okay? So, by adding the carbon dioxide onto the biotin, it activates the carbon dioxide. This is the first part, okay? Next, after we've activated the carbon dioxide molecule, this biotin carrier protein swings the biotin arm to the other side. So now the transcarboxylase is what's being active. You can see it's blue in color. This transcarboxylase will then transfer this carbon dioxide from the biotin to the acetyl-CoA. So if you transfer carbon dioxide to acetyl-CoA, you get a total of 3 carbons, and so you get malonyl-CoA. Okay? Was this clear? Okay, perfect. All right. Now, before we move on to step 3, which is the weird step of fatty acid synthesis, let's talk about how we regulate acetyl-CoA carboxylase, because we said it is the rate-limiting step and the regulated step as well. Okay? So, regulation in this ACC enzyme has 2 types. We have allosteric, okay, or covalent or okay. So allosteric means that something comes and binds directly to the enzyme. Okay. However, you can now guess that hormonal is indirect, okay? That happens through activating other things like phosphatases or kinases, and we'll see that, okay? So first, let's talk about the allosteric regulation of ACC. First, it is activated, okay, by citrate. So citrate stimulates ACC to polymerize and be more active. Now, how so? Didn't we say acetyl-CoA carboxylase is in dimers? Okay, so when it's not active, these dimers are separated. Okay, but when you activate them, they come together and they form a polymer, and then they activate. And I'll show you a diagram later. Okay, so why citrate? Because isn't citrate the reason why we're already starting all this? Okay, because we have too much citrate. We want to push it into making fatty acids, right? So citrate, like obviously, will activate acyl-CoA carboxylase to push the acyl-CoA into fatty acid synthesis. Okay? Now, by making fatty acid synthesis, the end product would be palmitate, okay, or palmitoyl-CoA. This will obviously inhibit acyl-CoA carboxylase. Okay, so this is a sort of feedback inhibition kind of thing, okay, because the end product will inhibit the process because, okay, I have too much fatty acids, please, like, stop making more of me, just stop, okay? So these two are allosteric, that means they go and bind to the enzyme directly, okay? On the other hand, the hormonal regulation involves insulin and glucagon. Okay, now these are involved in food, we know, glucose, okay? So insulin would activate acyl-CoA carboxylase through protein phosphatase, okay? But let's talk about why insulin. So usually when you eat, your blood glucose levels go up, correct? So when that happens, insulin increases to bring the glucose down. If we have too much glucose, that means insulin will be very high, and insulin will try to push the glucose or its products into storage, right? So it promotes fatty acid synthesis, okay? However, glucagon, which is produced in the other condition, when you're starving, for example, glucagon increases so that you break down glycogen to produce glucose, and then glucose increases in the blood. So you don't want to be making fat when you're starving, right? You want to be breaking that down. Okay, so glucagon goes to ACC and tells it, okay, please stop making fats, please stop storing my energy. We need the energy now. Okay? Now we'll talk about how exactly this hormonal regulation happens. But first of all, is it clear why each of these things activates or inhibits? All right, crystal clear. Okay, another concept before we go into the figures is that acyl-CoA carboxylase has two isoforms. Okay, so it has acyl-CoA carboxylase 1 and 2. 1 is the normal one we have. It's the one in the liver, mammary glands, adipose, and it makes fatty acids. Okay. ACC2, however, has an additional function, which is inhibiting CPT1, which is carnitine palmitoyl CoA transferase 1. It's a very long name. Okay. And this enzyme, or this, yeah, it's an enzyme. Okay? It's more of a channel, actually. It's needed for fatty acid oxidation. I will not explain that now because that is part of your fatty acid oxidation lecture or the beta oxidation lecture. Okay, but just know that ACC2 inhibits CPT1. Okay? Now, let's go through a diagram to explain this regulation. First, we said we have citrate goes to acyl-CoA to malonyl-CoA. And this step, acyl-CoA to malonyl-CoA, requires acyl-CoA carboxylase. Okay? First, we said, let's go through the allosteric inhibition. Citrate activates acyl-CoA carboxylase because obviously this is the starting product, right? So this is where you start from. So obviously citrate will push the enzyme to produce more fatty acids. However, if you have too much of the end product, which is palmitoyl-CoA, it will go back and inhibit the enzyme. It will say, stop making more of me. I'm done now. There's way too much. Okay? Now let's talk about the hormones. Insulin activates citrate lyase and mainly activates acyl-CoA carboxylase. Okay, we said because if you have too much food, insulin is a lot in the body and it wants to store this food. Okay, so it activates ACC and tells it, okay, there's a lot of food, please get rid of it, like store it. However, if you're starving or also epinephrine, if you're exercising, all of that, it's not time to store fat, okay? It's time to break down the fat or the glucose that you ate and to release the energy. So it goes to ACC and tells it to stop, okay? Now let's talk about the dimers and polymerization. First of all, the dimers, okay, here, they are in the inactivated state, okay? Meaning that they have a lot of phosphate molecules on them. Okay, so it is phosphorylated. Now let's take insulin, for example. Insulin wants to activate ACC. Okay, so it dephosphorylates it. how does it dephosphorylate it? By protein phosphatases. Look, it's written here. It activates ACC through protein phosphatases. A phosphatase is going to do what? It's going to remove the phosphates. So it's going to dephosphorylate ACC. So this takes it to the active state where there are no phosphate molecules on the ACC, and it is now in a polymer form, not a dimer form. Okay? The other way around, if you have glucagon, you want to inactivate it. So glucagon is going to phosphorylate acyl-CoA carboxylase. Now, how does it do that? By kinases. So glucagon or epinephrine inhibits ACC through pKa, or protein kinase A. You guys have heard about it a lot. Okay, so this causes the phosphorylation of ACC. Okay, and so you go in the opposite direction, and this is how acyl-CoA carboxylase would be in the inactive state. Okay, now I know that was a lot to take in, but is the regulation clear for acyl-CoA carboxylase? Because you will be asked about it. Okay. Okay, I'll consider that it's clear. Yes. Okay. So that was step 2. Now step 3, which is how we always see them, the weird elongating diagrams. Okay, so let's break that down. First, fatty acid synthesis consists of a series of 4 steps. So this step 3 or stage 3 has 4 substeps. Okay, these 4 substeps are condensation, meaning condensing the things together, reduction, which is adding hydrogen, dehydration, removing water, and reduction again, so adding more hydrogen. Okay, and they occur in this order. And we'll explain each one of them. Okay? Now, fatty acid synthase, this is the enzyme. Okay, this is the main enzyme in making the fatty acid chain. Okay? This is the enzyme catalyzing this reaction. There's an acyl carrier protein, ACP, on this fatty acid synthase enzyme, okay, on which the elongation of the fatty acid chain occurs. What do I mean by this? Fatty acid synthase itself has many things on it, okay? So it's not just a tiny enzyme, no, it's a big thing, okay? It's a big structure with many parts. ACP, or acyl carrier protein, is one of those parts on which the synthesis occurs. Okay, we'll see diagrams of this. But first, before we start synthesizing the chain itself, we need to link acyl-CoA and malonyl-CoA to the ACP end. So this is the reaction. Acyl-CoA plus the ACP goes to acyl-ACP, okay? And it loses the CoA group, and this happens through acetyl transacylase. Transacylase means that you're moving the acyl group. Okay, so transacylase. Okay, same way, malonyl-CoA with ACP, it's converted to malonyl ACP and loses the CoA through malonyl transacylase. Okay, this is just us getting ready for step 3. First part of stage 3 is condensation. Okay, so first we have this brown blob is the ACP. Okay, now you can see that malonyl is attached to ACP now. Okay, while the acetyl group is attached to this sulfide group. Okay, that's not important. Okay, condensation means that we basically take this acetyl group and add it onto the malonyl group, which is attached to ACP. So this would look this way. Okay, now count the carbons. You have malonyl is 3, acetyl is 2. Okay, however, after condensing, you lose a carbon dioxide molecule and look at the carbon numbers now. You have 1, 2, 3, and 4. So you've lost 1 carbon. So you went from these 5 to 4. Okay, this is important. Alright? So condensation, by the name, basically means that you condense 2 things together. You attach them to each other. So you take the acyl group, it doesn't have the CoA anymore, and you add it onto the malonyl group. However, 1 carbon atom is lost in this, and so you end up with a 4 carbon chain. Okay. However, this is not the end. of making this 4-carbon chain completely stable, okay? So this is in written form. Malonyl group is on the ACP end, or the brown blob, and acetyl group is on the sulfhydryl group, okay? Then this acetyl group is transferred to the ACP and it combines to the malonyl group. However, during this, you lose one carbon dioxide molecule, and so you end up with a 4-carbon chain, okay? So, is condensation clear? Good. Good. Alright, so we said we have condensation and then reduction. So, reduction, our main player here is NADPH, okay? Finally, this is where we'll be using it, okay? Even though we've produced it a very long time ago, okay? So, this is the 4-carbon chain that we have obtained from condensation. Now, I want you guys to notice that we have a double bond here on carbon number 3, or the beta carbon. Okay, so you count 1, 2, 3, and then we have a double bond. Okay, we don't want this double bond. We want to get rid of this double bond. Okay, so what we do is that we add hydrogen. How can we add hydrogen ions? Through NADPH. And this is a reduction reaction. Okay? Again, reduction means you add hydrogen ions. Okay, you basically spray hydrogen. Okay? So NADPH adds the hydrogen ions and it gets converted into NADP positive. Okay? Now look at carbon number 3. It's different now. It has hydrogen, and it has an OH group. Okay? So this is the reduction reaction. We've added hydrogen ions to get rid of this carbon, of this double bond, okay? And if you guys really want to know, this is actually a keto group, okay? So NADPH has a reducing power, meaning it adds H+, and this changes the double bond on carbon number 3, which is the keto group, to a single bond, okay? However, again, fatty acids are very picky. We do not want to keep this hydroxyl group, okay? So what do? We gotta remove it. How do we remove it? So we said condensation, reduction, then dehydration. So dehydration means removing water. You can see water coming out here. So you can take a look at this. We have HO and H, okay? And you have this carbon number 2 has 2 hydrogens, okay? Now look at what happened here. Carbon number 3 lost the hydroxyl group, okay? So that's HO. Now we need another H to make water, right? That came from carbon number 2. So here carbon number 2 had 2 hydrogens, but then it lost 1 hydrogen, okay? So in general, you don't really need to know these details, but in dehydration you've lost water, and you mainly got rid of this hydroxyl group, okay? This OH group, okay? So notice the OH group on carbon number 3. Here it is. In dehydration, water is removed. Now, this hydroxyl group is not there anymore. However, we have a double bond between carbon number 2 and 3. Fatty acids are picky. We don't want this double bond. We want to remove it. So how do we remove double bonds? By reduction. Okay? Remember, here in the first reduction, we removed a double bond, which was a keto group. Now here we have another double bond, so we will also remove it with reduction. Okay? Finally, reduction. So again, NADPH. NADPH adds hydrogen, okay, on this to get rid of that double. So look down here, carbon number 2 and carbon number 3, they have double bonds? No, okay? And they've gained a hydrogen. Carbon number 2 had 1 hydrogen, now it has 2 hydrogens. Carbon number 3 had 1 hydrogen, now it has 2 hydrogens. Okay? So now we have our saturated 4-carbon chain. Saturated, like we said, means there are no double bonds. Okay? And so this is where we've done the 4 steps needed, okay? Obviously, we don't stop at 4 carbons, we add more, okay? But this happens in a cyclical manner, all right? So let's read reduction first. To get rid of this double bond, reduction happens again with the help of NADPH. Now this double bond is removed, and we have the final saturated chain, okay? Now, before we move on, were these 4 steps clear? Condensation, reduction, dehydration, and then reduction? Okay, okay, good. Now, time to add more carbons, okay? So now we've made this 4 carbon chain, okay? However, in order to add more carbon on it, we need to move it around, okay? So we'll move this 4 carbon chain to the sulfhydryl group, okay, from the ACP end, okay? And then we will add a malonyl group on the ACP. So that's how we started off either way. We started off with malonyl on the ACP and acetyl on the sulfhydryl. But instead of having acetyl here, now it has 4 carbons. Malonyl stays the same on the ACP, okay? Then you get the whole cycle repeating again, okay? So the 4 carbon chain is now moved to the sulfhydryl group instead of the ACP, and the ACP is left empty. So what comes to fill it? The malonyl group, okay? Here you can see the malonyl on the ACP, and then the cycle begins again. So you get condensation where you move this 4 carbon chain to the malonyl group on the ACP, and then you get reduction, dehydration, reduction, okay? So you keep on doing this, okay, many times, and then you finally reach palmitate, which is 16 carbons, okay? Now, an important concept is how many times do I have to go through this cycle, okay, to reach palmitate, okay? So, does anyone have any guesses? Palmitate is 16, and we keep on adding 2 carbons out at once. So how many rounds do I need to get palmitate? 6, are you sure? 8, are you sure? Okay, actually, it's 7, okay? And it's not 8. Now, I know the typical way of thinking about this would be like, okay, 16 and you add 2 carbons each time, so 16 divided by 2 is 8, okay? But no, you already start off with 2 carbons, okay? So you don't count those. Let's explain this more. So this is palmitate again. We don't have numbers here, sorry, but this is carbon number 1, okay, 2, 3, 4, and so on, up to 16, okay? You already started off with 2, okay? So you add 2 each time. So again, this is the first addition, you end up with 4. Second addition, you end up with 6, 8, 10, 12, 14, 16. So you have went through the cycle 7 times to reach palmitate, okay? Now this is really important. How can we calculate this? There's a formula that you can use. It's the number of carbons, so here we have 16, divided by 2, which was how most of you thought, so 8. But, however, since you already started off with 2 carbons, you don't count those, so you minus 1. So you get 7 rounds, okay, for palmitate. And this is really important, okay, because most people think that it's 8. Most people just say, okay, divide by 2, but no, okay? You need to divide by 2 and then subtract 1 because you've already started with 2. These do not count, okay? Is this clear about how to calculate it? Then we start with 4 after the first cycle. Yes, but you added 2 to the acetyl, okay? So again, I'll just go back to, all right. So these are the 2 that I'm talking, okay? So these 2, okay, without the carbon dioxide. You already have this. You didn't need to go through the whole 4 steps to reach this, to be on the ACP, and it was already there in the beginning, okay? So step number 1 is to add 2 more from acetyl, and so you got the 4 carbon chain, okay? and then you added more. You added 2 more, so you got 6, and then 8. So let me count for you here. You already started off with 2 on the ACP end, okay? You did not need to go through the condensation, reduction, and all those to have 2, okay? You already had them, so these don't count. But to get 4, yes, okay, I did need to add the whole, go through the whole malonyl and acetyl, and then add that, and then condense, reduce, dehydrate, reduce. Okay, did you understand this part? Okay, good. Now, we are finally done with fatty acid synthesis, okay? But we're also going to talk about triglycerides, but this part is simple, okay? So, triglycerides, they are also known as triacylglycerol, TAG, or TG, same thing, okay? The structure is glycerol and 3 fatty acids, and we'll see figures about that. And these fatty acids are bonded to the glycerol by ester bonds. Okay, this is basic, like chemistry, biochemistry. Alright, so triglycerides can be synthesized by the body, de novo. Okay, what does de novo mean? It means synthesized de novo. Okay, that means you do not need to eat triglycerides. Now, triglycerides are in fatty foods. Okay, these are found in the diet, like we said, and something about triglycerides is that they are broken down into chylomicrons in the gut. Now, I think you guys have done your IT. You guys did your IT, yes. So you already know this. Okay, triglycerides are broken down in the gut into chylomicrons, and these chylomicrons carry them through the blood. Okay, so this is the structure. You have a glycerol molecule and 3 fatty acids. Okay, we've already seen how we synthesize the fatty acid chain. Okay, now let's talk about glycerol itself. Where do we get it from? Okay, glycerol has many sources. It can come from glucose. How can it come from glucose? Through glycolysis. We know that glucose goes through like glycolysis like 10 steps, and then you have dihydroxyacetone phosphate, and then you reach glycerol 3-phosphate. Okay, or you can have glycerol itself broken down from fat. Okay, so you have the adipose tissue, it's storing the fat, okay, and it stores the fat in the form of triglycerides. So when it wants to break it down, it breaks it down into glycerol and fatty acids. Then this glycerol gets converted to glycerol 3-phosphate by glycerol kinase. Okay, now this is not important. I'm just explaining so that you guys have an idea of, oh, where do I get the glycerol from? Okay, now you notice that I always say that glycerol has to end up as glycerol 3-phosphate. That is so that we can add the fatty acids. You cannot add the fatty acids to glycerol alone. It has to have the phosphate groups. Okay? And another source you can get glycerol 3-phosphate is by glycerol neogenesis, which means the making of glycerol, or from the T6. Okay? So again, glycerol 3-phosphate can come from glycolysis, glycerol kinase, or glycerol neogenesis. Not important to know these, okay? And then it combines with fatty acids to make triglycerides. Okay? Now, again, glycerol should be in the form of glycerol 3-phosphate, and it has many sources. Okay? Fatty acids, however, should be in the form of fatty acid acyl-CoA. Again, it's not fatty acids alone. It should have something with it so that you can add them together. Okay? So was this part about the glycerol and the fatty acids? Okay? Okay, good. So let's talk about the metabolism. So this is the same figure that was in your lecture. It's a good figure, okay? That's why I couldn't find another one. Right. So let's start off here in the adipose tissue, where one of the main sites where we make triglycerides. We have fatty acids that we've already synthesized, okay? And glycerol, okay? These combine to form triacylglycerol. However, in situations where you need energy, you need to break down these triglycerides. You can break them down by an enzyme called hormone-sensitive lipase. Okay, so you can tell that this enzyme is hormone sensitive Okay, so it reacts to hormones, and it's a lipase. Lipase means it breaks down fat. Okay, so triglycerides become glycerol and fatty acids. Okay, fatty acids are then transported through the blood, okay, to wherever they're needed to be broken down. Okay, and they can also go to the liver and then be stored there as triglycerides. Okay, you already have glycerol 3-phosphates in the liver because the liver has glycolysis, right? Glycolysis happens in the liver, so you can get glycerol 3-phosphates from there. Okay, now something else is that we talked about triglycerides being in food, and then you break them down into chylomicrons. So these chylomicrons transport the triglycerides in the blood. Okay, so and then they deliver the fatty acids to the liver. Okay, then the liver again, it combines the fatty acids with the glycerol and makes triglycerides. So from the guts, again, I'll just repeat this part. We've already taken triglycerides. We put them into chylomicrons. We take them to the liver. We break them down, and then we make triglycerides in it. Okay, it's like a whole thing. You don't need to know this in details. Okay, now the liver then packages these triglycerides into molecules or like kind of like packages. Okay, called LDLs or VLDLs. Okay, it stands for very low density lipoproteins. Okay, lipoproteins in general, whatever the types are, you guys have done GIT already, you know this. Okay, so they carry fats in them. They carry triglycerides. When they're circulating in the blood, okay, on the wall of the blood vessels, you have enzymes. Okay, one of these enzymes is lipoprotein lipase. Again, lipase means it breaks down the fat. So it breaks down the triglycerides in the chylomicrons into glycerol and fatty acids. Okay, now this was the whole thing. Like this is how triglycerides cycle inside the body, and I've written it in word form so that we can go through it once more. Okay, so in adipose tissue, so we're starting off at here, this yellow part. Okay, glycerol 3-phosphates and fatty acids combine to form triglycerides. We already know this. If energy is needed, so if I don't need to store the fats anymore, the triglycerides are broken down into glycerol and fatty acids by hormone-sensitive lipase. This one, okay? Then glycerol can go through many pathways, okay? It can go through glycolysis to produce energy, so it can continue the glycolysis pathway, or it can go in the reverse direction through gluconeogenesis, and it produces glucose. Okay, you guys already know this more than I do. Okay, the fatty acids themselves are transported through the bloodstream. Okay, if you really need to know, they're bound to albumin. Okay, so that's how they are transported through the bloodstream. They don't just float in the blood on their own. Okay, now from the gut, we've talked about how we have triglycerides in the diet, and they are broken down into chylomicrons, and then these chylomicrons are carried to the liver. So they carry the fatty acids to the liver. Okay, the liver itself synthesizes triglycerides, okay, and it packages them into lipoproteins. Now, these lipoproteins are thrown in the blood, and in the blood we have lipoprotein lipases on the endothelial wall. Endothelium means the wall on the inside of blood vessels, okay, and this enzyme degrades the triglycerides and it breaks them down to fatty acids and glycerol. Okay? So, is this concept clear about triglyceride metabolism? Okay, great. Okay, so now triglycerides are also regulated. Their synthesis is also regulated. Okay, so let's see how. Here we see we eat carbs, we eat proteins, okay, you get glucose, amino acids, and so on. Insulin promotes all these things to go into fatty acid synthesis, okay? So it promotes the conversion of glucose and proteins into fat, okay? So these would have to go through acetyl-CoA to fatty acids and through triglycerides, okay? However, If we have a patient that has type 1 diabetes, type 1 diabetes means that they don't have insulin, okay? So if you don't have insulin, what happens? You cannot promote this process, okay? So acetyl-CoA accumulates because it is not shifted into a fatty acid chain. So if it accumulates, the body decides to take it to another pathway, which is ketone body synthesis, which you will have in the next lecture about beta oxidation, okay? When ketone bodies are synthesized, okay, they cause many problems. So again, insulin stimulates the conversion of carbohydrates and proteins to fat, okay? In type 1 diabetes, where there is no insulin, fatty acid synthesis decreases. And so acetyl-CoA would accumulate and it would be converted into ketone bodies, okay? So sometimes patients have this attack, okay? It can be called ketosis, severe ketosis, or diabetic ketoacidosis, okay? You'll hear it as DKA, okay? How do these patients present? You can smell their breath and it would smell like acetone or alcohol, so they might be mistaken for being drunk, okay? Even though no, they're actually having a crisis. Okay, and this can be very fatal because ketone bodies are actually acidic, so they lower the pH of the blood and they can kill the patient. Okay? Now, one more thing about glyceroneogenesis, which is basically the formation of glycerol. Okay? We've got it here. Now, you guys know this pathway. This is glycolysis. Okay? So, pyruvate, oxaloacetate, blah, 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 okay, to glycerol 3-phosphate. However, you can notice the star here between oxaloacetate and phosphoenolpyruvate, and this star marks the enzyme pepcarboxykinase. Why are we talking about this? Because there's a drug that we use in type 2 diabetics, okay, called glitazones. And these basically stimulate pepcarboxykinase to convert oxaloacetate into phosphoenolpyruvate and go down and produce glycerol 3-phosphate so that you can make triglycerides. So again, glyceroneogenesis is the synthesis of glycerol. It can happen through glycolysis, okay? Glitazones are the drugs that target pepcarboxykinase and induce its activity, and so this promotes the synthesis of triglycerides to decrease the free fatty acid levels in the blood. You don't want fatty acid levels to be roaming around in the blood in diabetics, okay? So, yeah, before we move on to the questions, was the part about triglycerides clear? The whole thing. Do we need to know the name of the drugs? So it's just this one drug, glitazones. Okay, this is the only one you need to know. Okay, yes, you do need to know it. Okay, but it's just one drug. Any other question? It's great. Okay, now we will be going through practice questions, and I need you guys to tell me what the answer is. Okay, so first practice question. All of the following statements about acetyl-CoA carboxylase are correct except. So you guys tell me what you think it is. Okay. Someone's saying D. Okay, D. Okay. Does anyone else want to answer? More answers? D because it's inhibited by it. Good. Good job. Okay. So yes, it is D, and as Rizan explained, because palmitoyl CoA actually inhibits ACC. Okay, because it's an end product of fatty acid synthesis, so it would inhibit the entire process. Okay. Now, second question: the end product of cytosol fatty acid synthetase in humans is D. Okay, D. D. Okay, good. So we're all on the same page. Yes, it is D. It is palmitic acid or palmitate. Why is that? Because we said fatty acid synthase can only make carbons up to number 16. Okay. Then we get stearic acid through an elongase enzyme, which is 18 molecules. And then you get oleic acid, which has a double bond at carbon number 9 through a desaturase enzyme. Okay. Now, third practice question. Now this might be a bit complicated. Let's see if you guys can do it. So let me read it. The rate of transcription of the mRNA of which of the following enzymes is likely increased in an individual on a fat-free, high-carbohydrate diet. C, it's giving C. Alright, no. Okay, not B. Okay, so it is A. Why is that? Okay, because we said that this person, let's look at the person's diet, okay? The person is on a fat-free diet, okay, meaning they do not take in fats at all, okay? However, they are on a high carbohydrates diet, meaning that they're taking in too much sugar. If you take in too much sugar, you will want to store it as fat, okay? So what is the enzyme that makes fatty acids to store them? It is fatty acid synthase, okay? Now, why is it not C? Hormone-sensitive lipase is basically involved in breaking down the fats, okay? However, you're eating a lot of carbohydrates, right? So do you want to break down this fat? No, because you don't have fats anymore. You actually want to store it, the excess energy you're taking in, okay? Is that a question? Oh no, it makes sense. Okay, last question. An increase in which of the following will most likely increase mobilization of sorry, of fatty acids from fat cells? Hello, hello. Now it's B. Good. Okay. Now it is B. Hormone sensitive lipase activity. Because we said mobilization of fatty acids from fat cells, meaning that we take fatty acids out of the fat cells. Now, what is the enzyme that breaks down the adipose tissue, or basically the triglycerides in the adipose tissue? It is hormone sensitive lipase. Okay. So, good job, you guys. And thank you so much for joining the session today. I know there aren't a lot of people, but thank you either way. And please do scan this QR code because the feedback. And this really was a long chain of events. Mhm. So do you guys have any questions before we end the session? Thank you. Thank you. Okay then. You're welcome. You're welcome. I think we can end the session now.